Okay, it has started. So I was telling that today we will study about the SDS page. The full form of SDS page is sodium dodecyl sulfate polyacrylamide gel electrophoresis. Another common name of it is SLS, sodium lauryl sulfate polyacrylamide gel electrophoresis. So basically this is a separation technique which is used for the separation of a mixture of protein based on the molecular weight or the size. As we have already discussed with you about the gel electrophoresis, which we have used for the separation of DNA with the help of agarose gel. So the same pattern is used for the protein separation. The basic idea and principles are same. So here also we will use a matrix which is in the form of gel, and here it is polyacrylamide gel. We will load our fragments of protein into it, and then we will encounter electric current, electric field around them, and the movement of proteins will take place from the negative to the positive terminal on the basis of their molecular mass or molecular weight. So this is the basic principle. So each and every point of how the gel is prepared, what's the different type of system we are using, we will see it in a great detail. So first let me tell you how the sample preparation takes place. So for the sample preparation, we use the protein or the protein mixture which we want to separate along with a denaturation buffer. This denaturation buffer is also known as sample buffer or SDS page buffer. The component of this denaturation buffer is very important. So first is the tris chloride. So this chloride has a negative charge. So why it's used, we will see in the later part of the presentation. Second part of this buffer is beta mercaptoethanol. Then there is SDS, then glycerol. Glycerol provides the density. Then there is a bromophenol blue staining dye and double distilled water. So why we are using double distilled water is that we don't want any ion to interfere during our protein separation. So now the most important two parts of this buffer is the beta mercaptoethanol and SDS. So we will first discuss about it. So we all know that protein have the protein is basically made up of different type of amino acid. So there is no fixed charge on a protein, and also the protein is quite complex and it is in a folded state. Most of the proteins are in the folded state. So the first objective of this gel electrophoresis is we want to unfold the protein, and the folding of the protein is basically because of the disulfide bond which is present in between them. So how we will break this disulfide bond? Here we will use a reducing agent, that is beta mercaptoethanol. So this beta mercaptoethanol will break the linkage within the protein and separates its subunits. Here we will achieve almost a primary structure of the protein. But there is another difficulty, which is the protein has different type of amino acid joined in a single chain. But amino acid also have different charges, positive, negative, maybe polar or non-polar. So separation can't be done. So we need some agent which can impart a common charge on the entire chain. And here comes the picture of SDS. So SDS is basically an anionic detergent which binds to the hydrophobic region of the protein, imparting negative charge to the entire protein structure. Now, suppose we have three different type of proteins. One is small, another is a bigger one, and the last one is very small. So, when SDS is applied to it, so negative charge will be imparted on the entire fragment. But the negative charge is directly proportional to the number of amino acid. So, larger the protein fragment, more negativity it will have. Now, if we look at the third point in this slide, So if we see the third point in this slide, so we can see that SDS binds proportionally to the size of the protein, and approximately 1 molecule of SDS binds to every 2 molecules of amino acids. So after they impart total negative charge on the chain of the protein, now the separation will take place by the help of molecular weight or the size. This was the basic principle. Now here we will talk about the gel in detail, because the gel is different from the separation of DNA which we use agarose. So polyacrylamide is basically a synthetic polymer, or it's like a chain of acrylamide monomers. Now to join this acrylamide monomers into a cross-link or a mesh-like structure, we need the addition of bisacrylamide during polymerization reaction. Now the polymerization also act in presence of an initiator and a catalyst. So the initiator is ammonium persulfate and the catalyst is TEMED, that is tetramethyl diamine. Once the mesh is formed, there are pores. We will load our protein into it and the separation will take place. Now, this acrylamide, for the formation of this mesh, the concentration can vary from 10 to 15%, and the amount of proteins which can be separated is 12,000 to 70,000 daltons. Here is the it's in this slide we can see how it's happening. So in the B picture we will see first. So there is two protein subunit A and B, and they are linked with disulfide bond, whereas in the C picture we have only a single subunit. Now we will heat it with SDS and beta mercaptoethanol. So what will happen, the disulfide bonds will break and a total negative charge will be imparted. Now we will load the thing into a polyacrylamide gel electrophoresis gel, and we will say that because here there are two units of protein, so we can see two bands in the gel, whereas in the single subunit we can see only a single band. And the upper band is of the B1 because it's like longer, so it will travel less, whereas the A will travel the most. So here we can add one more point that I have already told you, the reducing agents which we are using here are SDS and beta mercaptoethanol, but we can also use DDT. So here again a picture is given how the loading is done, how the migration is taking place from negative terminal to positive. This you will see the entire video in the later stage of the presentation and you will get a clear picture. In the B diagram, you can see here is also a ladder and there is an unknown protein, and from that we can find the approximate molecular weight of our unknown protein. Here is the apparatus of SDS page. So we will see the video and I think you will get it more clearly, but I will tell you just that here are glass plates in which we cast our gel. Then there is the frame in which we place the glass plates. Then there is a comb for preparing the wells, and this is a casting strand. It's the frame and electrode assembly, and this is the tray. Now let's see gel preparation. So in SDS page, what happens is we use two type of, it's not a continuous gel, it's a discontinuous gel. That means it has two sections. So I will draw here how it's happening. Sorry. So you can see the gel. Wait a second. So suppose this is our gel. So this gel has two parts. The uppermost part is the stacking gel and the lower part is the separating gel. So what's the difference between stacking gel and separating gel is that the pH of this part is around 6.8, whereas the pH of the lower part is around 8.8. So what happening is, first of all, as I have told you in the earlier sample preparation part only, that in that we have protein along with a tris chloride. So this tris chloride has a negative charge. So we will write what are the components. So first of all, we have tris chloride, which have a negative charge. Then there is a SDS plus protein complex, which is also negative charge. Now we place this gel into a tray and we pass a buffer from the above. This buffer is known as migrating buffer. So this migrating buffer has another very important component, that is tris glycine. So why I am telling you this glycine is very important in entire SDS component. Now this glycine is the simplest amino acid because it has only hydrogen in its side chain. Now this glycine is a zwitter ion, and we know what is a zwitter ion. So zwitter ion, that is it at a pH of 2.3, this glycine is positively charged, that is protonated, whereas at 9.6 it is negatively charged and deprotonated. If we plus 2.3 plus 9.6 and divide it by 2, we will get 5.95, and at this pi value the glycine is neutral. So whenever we move from 5.95 to 9.6 pH, our glycine gets slightly negative towards the 9.6 pH. So why this is important and how it's functioning, we will study one by one. So let's start with, first of all, the stacking gel.
Ma'am, so what's the relationship here with Jupiter and pH?
Yes, we will see once you see the entire thing, and if you don't understand, then I will clear it off again. Okay. So let's see first of all this stacking gel. Now stacking gel, as I have already told, that has a pH at 6.8. So now there are three main components. This glycine, this chloride, tris chloride, and our SDS protein complex. So chloride has a negative charge. SDS protein also has a negative charge. But glycine at this position, because the pH is only 6.8, it is either neutral or either slightly negative. Okay. So here comes the picture why we are talking about zwitter ion. Because it is 6.8, we have considered glycine either slightly negative or towards the neutrality. Now when we will add our sample, so what will happen, chloride, because of the highest negativity, will go at the bottom. We will highlight it with a different color. So it will go here. This is our chloride. Okay. Then because SDS with protein complex is also negative, so it will form the second layer. And Lastly, because glycine is having the least negativity, because it is 6.8, it will come very slowly and it will push the protein in between the chloride and itself and form a sandwich. So why we need a sandwich or why we are stacking the protein is because we don't want that the protein, if the proteins will be randomly separated or randomly present in the gel, then some protein will move fast, some protein will, the molecular weight won't be crucial for their separation, because randomly placement will cause random separation. So at this point, we want that all the protein should be sandwiched or stacked between two important ions, that is chloride and glycine. Okay. Now, because here one more very important thing which I would like to tell you is, at stacking gel, the gel concentration is only 5%. So the pore size will be very high. So the movement of protein will also be faster. So they will form a sandwich easily. Now comes the picture of separating gel. So we will see how it's working. So it's our separating gel. Now this separating gel has a pH of 8.8. Okay. Now at this, chloride will be negatively charged as usual, and the SDS will also be negative with protein. But here the glycine will also be highly negative. Why highly negative? Because the pH is 8.8 and its maximum negativity is at 9.6. So what will happen at this position, chloride, because of its highest negativity, will be present here only at the bottom. But this time glycine, because its negativity increases, it will be present second. And lastly are proteins. So because the protein were stacked together, the protein will always in a line or will be in a singular plane. Now what happens here is, because this gel is made up of polyacrylamide at a concentration of 12%, that is very high, so the pore size will be very small. So now the separation of the protein will take place on the basis of molecular weight. Because the protein are in a singular line, so smaller fragments of protein will migrate faster towards the positive pole, whereas the larger ones will diffuse slowly. So here is the understanding and the basic principle how the two gels are functioning in SDS page. Now, in this, the two composition of the separating gels are written. First is the separating gel, the second is the stacking gel. Stacking gel is always present on the top, whereas the separating gel is present at the bottom. So we use distilled water, then we put acrylamide solution in both the cases, but the percentage are different. Then we use Tris, then SDS, APS, Stimit, all are the same, both in separating gel and stacking gel, only the concentrations are differing. So we will allow the polymerization and we will see how it's functioning. We will see the video, and if you don't understand, you ask me then. Now once the proteins are separated, now we want to visualize them. So how we will visualize them? We will visualize them by a dye which is known as Comasi blue. Now the Comasi blue can be detected at a very low concentration of about 0.1 μg of protein per band. So what we do, first of all, we take our gel out of the casting tray and then we put methanol, water and acetic acid along with our Comasi blue dye into it. And we leave it for 4 to 5 hours so that the staining of the dye can take place. Once it is done, we will destain the dye because we don't want excess dye to remain in the gel. So we will again destain it by using of methanol, water and acetic acid. Once that is done, we will visualize the gel. Here is the sample preparation for this experiment particularly. So here we have used actually three type of protein, that is BSA, lysozyme and oval albumin, but we have not used in the later part. We have only used BSA and lysozyme, which you can see. Then here also we have produced that denaturating buffer I have told you, or SDS page buffer, in which we have used stress chloride, we have used SDS, beta mercaptoethanol, bromophenol blue, and we will also use glycerol in this also. So then we will take a water bath and boil it for 2 minutes and centrifuge to mix it properly for 1 minute. Then we will prepare our gel, we will put the comb, we will remove it, we will load our material, we will transfer it to the running tank, and we will allow the sample to run under the influence of electric current. So when the gel runs through the stacking gel, the 25 milliampere is applied, but when it is in the resolving gel or the separating gel, it should be 35 milliampere. So as in the previous slides only we have discussed that the staining of the gel is done by Comasi blue staining dye. So here only the composition of water, methanol, acetic acid, that is 5:4:1 given, and we use only 0.1% of Comasi blue. Then the destaining of the gel is done, only the Comasi blue part is not there, except the previous composition or the ratio remains the same. And finally we take the photograph of the gel using a gel documentation unit. So now we will see how is it happening. Okay. So with this we are pouring all the dissolving gel components together, and because the resolving gel or the separating gel is at the bottom, so we will fill it first. Once we see in the tube that the resolving gel has solidified, we will now fill the stacking gel. Here we are putting all the components of the stacking gel together, and now we will pour it to the tray. Once it is also solidified, we will take out the tray and we will put it into the chamber where we will put the buffer and start the process. we will remove the comb and then we will add the ladder, and then we will also add the protein of our interest which we want to know, target protein, along with the bromophenol blue dye, and then we will allow it to run. Once the gel run by 3/4 part, we will turn off the power supply and we will take out the gel gasket. Now this is the process of staining of the dye. Staining of the gel. And we will leave it for 3 to 4. Once that is done, we will wash it with molecule water and then we will put the tea stain solution for the overnight test. And after that, we will observe something like this. So on the first part, on the very left side, we see the protein ladder. Then because here we have used two type of protein, that is lysozyme. So and BSA, we can see its bands. So lysozyme is around 11 kilodalton weight, molecular weight, and BSA is around 72 kilodalton. So that's all from my side. If you have any question, please continue. Can you hear me?
Yes, ma'am, we can hear you.
Okay, so if you have any questions, please, Ratnesh, you were asking something.
No, ma'am, not. I have not any question now
Okay,
today.
so then if you are not asking, then I will ask some questions to all of you. So, in the previous class, I have taught you about that we have used the agarose gel electrophoresis. So that was an